Good evening, everyone. We'll wait for 5 more minutes to see if others join, and then we can start. Good evening, everyone. I hope you can hear me. I'll just share my screen. Can you all see the presentation?
Yes, ma'am.
Okay, yeah, thank you. So, good evening, everyone, and welcome back to week 9. We have been talking a lot about drug delivery, and these are the PMRF sessions for NPTEL online course, Drug Delivery Principles and Engineering. So, as I said, we have been talking a lot about drug delivery and tissue engineering, and let's go over some of the points that were discussed in week 9 lectures. So, the foremost was, as you might remember, for the past few weeks, we have been talking about different routes of drug delivery. So, we have discussed oral route, which is the most conventional and the most patient compliant route, but it has its own disadvantages, as you have seen that it suffers from first pass metabolism, or if your patient is unconscious, you cannot give a drug, you cannot administer a drug orally. Then we have different routes such as subcutaneous and intramuscular. These are the parenteral routes, meaning they avoid the first pass. They avoid the gut, the flowing of the drug through the gut system, and in that process, they then become advantageous to overcome the first pass metabolism effect. And although these are injectable, so you have to give certain training to the patients if they're going to self administer the drugs. And then next we have transdermal route. So this is nothing but delivering your drugs through the skin. So although it is a very patient compliant route, it has its own disadvantages, wherein we saw that if you have a lot of there's a lot of barrier for the drugs to be transferred from your system to your to the blood because of the dead skin layer which you have. So there are different ways of again overcoming that. We saw there are few chemicals which you can apply to destabilize the layer. And then talking about, through the drug delivery perspective, we saw that we can make microneedles out of polymers. So these are nothing but needles of around 300 micron length, which you then put a patch of these needles on the skin. So these needles will then since they're sharp enough to destabilize the layer, but they don't go as deep as where your nerves are, so the patient won't feel any pain, but then you are making a very efficient drug delivery system through this process. Next, we discussed intra-articular. So this is nothing but delivering drugs directly to the joints. So it becomes most prominent in case of knee joints, for example, for the treatment of osteoarthritis. So this is a very specific, a trained personnel has to do it. Patients mostly cannot self-administer these type of routes. And then lastly, we discussed about intravenous. So this is also a very popular drug delivery route. So you must have seen this being used a lot in hospitals where people get IV drips. So this is a very advantageous route, considering if you want immediate drug, the effect of the drug to be very immediate. So in that case, we can go ahead with intravenous route. But then we discuss the disadvantage also of this route, because now everything is directly being injected into your blood. It will go all over the body. It might have non-specific effects, and also there are more chances of getting infections because of this. So this was all about drug delivery routes. In this week's lecture, a different module was being introduced by the professor. So this is so henceforth we are going to discuss about immune response to materials for a few lectures. So as you might remember, in the initial sessions, we saw this effect of, let's say there is something foreign being implanted into the patient's body, then naturally, because our immune system has been evolved for so many years now to detect foreign material and to act against this, to eradicate the foreign material. So naturally, whatever you're going to implant or whatever you're going to deliver in the patient's body is definitely going to elicit some immune response. So in that case, let's say you have put an implant in the patient's body, so you want the drug to be delivered from this implant for some other disorder, for some other disease the patient has. But now the patient's body is going to consider this as a foreign material, and it's going to start attacking this implant that you have put in the patient's body, and it will try to eradicate. So this will then lead to constant inflammation in the patient's body, which is not a very good state. So instead of now treating the disease, you have again given some more problems for the patient to deal with. So this is not an ideal case. So in order to reduce this immune response, which is going to be elicited by the human body against the materials that you are putting inside, so we first need to understand how the immune system works. So a lot of detail about the immunology was discussed in this week's lecture. There were a lot of cells about immune systems that were discussed. So I'll just briefly go over a few points. So immune system is basically divided in two branches. One is the innate immune system and one is the adaptive. So innate immune system is non-specific and the immediate effect that happens. So it's the first thing that comes up whenever the body encounters any foreign material. So various different cells like macrophages, your neutrophils, eosinophils, basophils, they form a part of this innate immune system. And then over the course of time, then your the cells of your body, the specific cells which are your B cells and T cells, they then get time to adapt to the foreign material that has entered. Let's say there's a pathogen which has come up and your body has never encountered that pathogen before. So in that case, your body will need some time to learn what that pathogen is and how to best eradicate that. So in that case, then this is where your adaptive immunity will kick in. And so after a while, so this is very specific, so whatever pathogen has entered, your adaptive immunity is going to be very specific against it. And because it's taking time to learn what the pathogen is, it has a late onset and then it stays for a very long time. For example, your B cells, the memory B cells which are generated whenever a new pathogen comes in, they will circulate in your body, they will stay in your body for even years. So whenever next time the same infection happens, your body is very ready to fight against that infection. It doesn't need much time to learn about that. So this was about innate and adaptive immunity. One important part of immune system is inflammation. So we need to understand this process in detail. So inflammation is nothing but a cascade of events. It's a series of events. It happens one after the other. So what exactly happens is whenever there is an injury to the body, for example, while putting an implant, maybe you ruptured some of the blood vessels, or let's say there is a foreign material that's coming. It might be a pathogen, it might be your drug delivery system, anything. Whenever that thing enters, for example, let's consider there's a pathogen that has entered. So as I said, the cells of these innate immune system, which are your macrophages and neutrophils, they are always circulating in your body, and they're always, a lot of these macrophages are also present in the tissues. So whenever a foreign material enters, let's say the pathogen is circulating in the blood, in that case your macrophages are going to identify that this is a foreign material, and they are very phagocytic cells. Phagocytic meaning they engulf everything that is foreign to them. So they are going to take in the bacteria, and then this will lead to immune cell activation. So your macrophages are going to digest this bacteria, and in this process, because it knows that this is foreign, the macrophages are going to release something known as cytokines. So cytokines are nothing but proteins. These are signaling molecules. They will then alert your body that there is an infection and we need to act ASAP. So these cytokines then will attract a lot of different blood cells. Let me just take a pointer. Yeah, so we are here. These cytokines will then, as I said, these are signaling molecules, so they will signal to the other cells of your immune system. They will then call in a lot of these leukocytes, these white blood cells, and then eventually these cells will then take care of that pathogen, and the body will then go towards the healing process. So this is inflammation in short. So this is discussed in detail in the lectures as well. You guys can follow it later on also. And another important part of the immune system is the complement system. So the reason why we are learning all of this is because it's going to be very relevant once you introduce biomaterials in a patient's body. So let's say we were discussing about this complement system. So as I said, this is part of innate immunity. So it's a non-specific immune system immunity here. So what exactly is this complement system? It's nothing but these are a series of proteins, specifically 20 proteins, which are in their unexcited state. They are very inactive. They are just circulating in your body. The moment so their main function is to clear microorganisms. So the moment they encounter any microorganism, these then become active. So as I said, these are a series of small proteins, these 20 plasma proteins. So first of all, one molecule will identify that there is a pathogen, and then it will keep on triggering the next molecule. So in a way, what this complement system does is it will identify the microorganism. There are a lot of functions of these complement system, but most important is it's going to highlight that antigen. It's going to alert your body that there is an infection in the body, and the body needs to clear it. So there are different ways through which this complement system gets activated. There are specifically three. One is the classical pathway, one is the alternative pathway, and then there is third, that is lectin mediated pathway. So in case of, so since we are learning about what happens to biomaterials, the most relevant complement system activation in case we are looking at biomaterials is the alternative pathway. So what happens in a series is, once you have your material implanted in the patient's body, no matter where you put it, let's say you have put it on the skin, but sooner or later that is going to come in contact with your blood, because blood is flowing everywhere in the body. So once the material comes in contact with the blood, as I said, these complement system proteins are flowing in the blood in an inactive state. The moment they encounter this material, they're going to be activated. They then what they do is they highlight the particular whatever material is implanted, and they alert the body that there is an infection and we need to take care of it. So this increases the immune response. And in case while implanting your material, let's say you have ruptured few of the smaller capillaries, so this will then trigger the blood clotting cascade also. Your body will think that there is some injury inside the body, and it will start repairing that particular injury. So now what happens is, since there is a constant inflammation in the body because of the material implantation, a lot of immune cells are coming in and attacking your particular implant. So this will lead to device failure. So the only option to save the patient from this is you have to remove that device. So in that case, your therapy will fail. So that is why it becomes important to understand how the immunity of the body is going to react against whatever you have implanted. So there are ways to reduce this complement activation. So now you must have understood that we need to reduce this complement system activation. So there are different ways to do that. So we'll see some of these in the subsequent questions as well. And the last point that was discussed in week 9 lectures is the adaptive immunity. As I was talking, that adaptive immunity is very specific, and although I said that there are two branches of immunity, the innate and adaptive, they are not very separate, they are interconnected. So whenever there is a pathogen present in the body, as I said, your macrophages and your neutrophils, they're going to engulf this, they're going to phagocytose this pathogen, and in the process of killing the pathogen, they will then chop up this pathogen. And there's a process known as antigen presentation. So you'll get the details in the lectures as well. I'm just introducing it briefly here. So as I said, the macrophages will chop up the antigens, and what they do is they present the antigens on their own surface. Presenting in the sense, they'll just take an antigen, put it in its membrane, macrophage membrane, and they'll just decorate the surface with these antigens, just to let your T cell and B cells know that this is the particular antigen you're supposed to fight against. And in that process, then T cells become activated. So your cytotoxic T cells will then identify that this is a particular antigen, I need to kill this microorganism. And then your B cells will then produce antibodies against this particular antigen. So whenever the pathogen comes again, that pathogen is going to carry that antigen, but your B cells already know that it's supposed to produce antibodies against this, and in that process they then clear this off. So these were the points that were discussed in this week lectures. So if you have no doubt in this, then we'll move on towards the questions. If not, if somebody wants me to explain any of these concepts in detail, then we can go through this again. For now, I'll just move on towards the questions. So the first question in the series is, which of the following options represent the correct chronology of events in inflammation? A lot of options are given. I suggest you take some time to read through these, and then if anybody knows what the exact sequence is, you can let me know. We just discussed some of these very briefly in the introduction. So if anybody has the answer, you can let me know. Anybody wants to volunteer?
Option 7.
Are there two people talking? I'm sorry, I didn't catch your response. Can you please repeat?
Option 2.
Option 2, okay. Anybody else? Thanks for answering. No? Okay, let's look at the answer. The correct answer is actually option 1. So as I said, inflammation is a cascade of events, right? So there are specific events happening one after the other. So what happens first is, whenever there's a tissue injury or any foreign entity present, your tissue macrophages are going to become activated, and they're going to secrete a lot of cytokines. So these cytokines also have a very specific function. What they do is, they first of all will undergo they'll cause a process known as vasodilation. This is nothing but increase in diameter of the capillaries. Now, since the injury that has happened, or the pathogen might be present in the tissue, and all your immune cells are flowing in the blood vessels. So what the immune cells have to come from the blood to the tissue, right? So to do that, the cytokines will then cause vasodilation. Vasodilation is increasing the diameter of capillary, so that more and more of your immune cells will come to the tissue. So that is the first process that happens. Next, it increases capillary permeability. As I said, your blood vessel and your tissue. So your cells have to flow from the blood vessel and they have to enter the tissue. The blood vessel is always lined by endothelial cells. So usually the endothelial cell lining is very very tight. They will not allow anything to enter from the blood vessel to the tissue until unless they get the signal for it. So now that they have gotten the signal that cells have to enter from the blood vessel to the tissue, the cytokines will cause capillary permeation. So a lot of fluid will then flow from your blood to the tissue. This is known as edema, the swelling of. So whenever, you must have noticed, whenever there's a mosquito bite or some injury, the inflammation kicks in and you can feel that the part where the mosquito has bitten, it's swollen a little, it has become red and it's kind of warm. So all of these processes happen in inflammation. So first vasodilation will happen, then your edema will happen. The fluid has to flow from blood vessel to the tissue, and in that process most of your leukocytes, there's an influx of leukocytes, a lot of your white blood cells will come to the tissue, and then they will act against whatever injury is there. They'll start repairing it. If there is a pathogen, they'll start engulfing and killing it, and then later on it will lead to healing and tissue regeneration. So after a point, your inflammation has to stop. If there is a constant inflammation, it will start killing all your it will start harming all your normal cells also. So after a while, whenever the injury is repaired, your body also stops the inflammation cascade and it starts regenerating the particular tissue. So the correct sequence for inflammation is vasodilation, edema, influx of leukocytes, clearance, and then healing. Is this clear? Okay, no doubts. Then let's look at the next question. Next is the three arms of complement cascade converge at a protein complex which is known as, and then you have your options. Any volunteers?
Option 2.
Option 2 is C3 convertase. Okay, thanks for answering. Anybody else has a different answer other than option 2?
Option 2, ma'am.
Okay, the same answer. Thank you. Okay, let's look at the answer. Yeah, both of you are correct. It's C3 convertase. So as I said, your complement system has various different pathways through which it can get triggered. So the first is classical pathway, alternative pathway, and then lectin mediated pathway. So no matter how your complement system is going to be activated through all these pathways, they're always going to converge at one particular protein complex, that is C3 convertase. So no matter if it started from classical or the alternative, it might start from different molecules getting activated, but ultimately they have to somewhere converge at C3 convertase. So this is the common protein complex in all the three pathways. So yeah, that's your option 2. Please let me know if I'm going too fast or if I'm talking very fast so I can slow down a little. Okay, let's look at the next question then. Next is, which of the following approaches can be employed to reduce complement activation on a biomaterial surface? And you have to choose two correct options. Any volunteers?
Option 5 and option 2.
Option 2 is reduce surface nucleophiles and 5 is heparin adsorption to the surface. Okay, thanks for answering. Anybody else? No one? Okay. Yeah, you're perfectly right. The correct answer is reduce surface nucleophiles and heparin adsorption to the surface. So as I said, since this complement activation is not good when you want to implant something in a human body, so there are different ways to reduce this. So the first is you have to make the surface less nucleophilic. So so C3b is one of the proteins in this complement system cascade. So this C3b has a tendency to attach itself to a nucleophilic surface. Nucleophilic meaning a negatively charged surface. So if let's say this is your device and this is the membrane, your drug is going to flow from here to here, and your membrane has a lot of negative charges now. So your C3b finds it very compatible to go and attach itself to the surface. In that process, this will get activated, and then subsequently this will start activating other proteins of the complement system, which will then cause a lot of problems, and it will then lead to a lot of immune reaction. So the one way you can reduce this complement activation is make your surface less nucleophilic. So that's what we have here, reduce surface nucleophiles. Another way is to adsorb heparin to the surface. So what heparin is a naturally occurring molecule. It's found in your ECM, the extracellular matrix. So heparin along with another protein which is factor H, it will work together to inactivate the C3 convertase. So the C3 convertase is a very important molecule which then activates, it's an enzyme basically, which then activates the downstream molecules in the complement pathway. So to avoid that, to inactivate C3 convertase, we can just coat this membrane with heparin, which will then work with factor H to reduce this C3 it will cause inactivation of the C3 convertase and then reduce the complement activation. So that's why the answer to this question is, if you want to reduce complement activation on a biomaterial surface, you can reduce the surface nucleophiles, or you can you can adsorb heparin on the surface. Is this clear? Okay, I'll move on towards the fourth question then. The next question is, which of the following factors can cleave fibrin in vivo? And we have various different options here.
Plasmogen.
Okay, one response is plasmogen. Anybody else wants to give it a try? Thank you. Okay, somebody is answering option 3 in the chats, which is fibrinogen. Okay, thanks for responding. Anybody else? Okay, let's look at the correct answer then. The correct answer is actually plasmin. So as I said, what exactly is fibrin? So what was the question? Yeah, can cleave fibrin. Okay, so as I said, if you have any injury while implanting your material, if there is some rupture of the smaller capillaries or any blood vessel gets ruptured in the body, your body will trigger a clotting pathway, the blood clotting pathway. So what it does is, it will activate a lot of factors which will then cause this blood clot. And after so this also happens in a normal situation also, where you must have observed that if you have a cut on your skin, in a few days you will see a scar tissue forming on that, a black thing forms, and then later on your body automatically will keep on resolving, if you don't pick it of course. So it will keep on resolving that clot, and then a new skin will form over that injured part. So this clot is basically made of fibrin. So to dissolve this fibrin, there are actually various different proteins in this cascade. One cascade is where your clot forms, another cascade is where your clot dissolves. So to dissolve this fibrin in vivo, the factor that is responsible is plasmin. So this is a very complicated pathway. So if you start looking at it one by one, you'll see that for formation of the clot, you have to follow this particular pathway, wherein your different factors will first activate your thrombin, which will then help in formation of this clot. And then if you want to dissolve it, then your fibrin will kick in, and your fibrin then acts on the clot and then dissolve the clot. So the factor responsible for dissolving the clot is your plasmin, which is then in turn activated by plasminogen. So as I said, it's a cascade. One molecule will activate the next, then the next. So in this process, the clot will be dissolved. So the correct answer to this question is fibrin is cleaved by plasmin. Clear? Okay, let's look at the next question then. Next is, pick the incorrect statement regarding cytokine. We have to choose two options. I'll give you some time to read through the options, and then if anybody knows the answer, please let me know. Anyone wants to give it a try?
Option 3 and 5.
3 is cytokines can act in a paracrine manner, so that cytokines affect the behavior of adjacent cells. And 3, 4, 5. Cytokines are highly potent proteins that bind to their cognate receptors and induce downstream signaling cascades in the respective cells. Okay, somebody's answered 3 and 5. Anybody else? Thank you for responding. So please read the questions carefully. It's asking us to choose incorrect statements. So for this, you need to know what cytokines exactly do. No more responses. Okay, let's look at the answer. So the incorrect statements about cytokines are these. Cytokines are small lipid molecules that are released by various cells in the body, usually in response to an activating stimulus. And cytokines in their active forms are always pure peptide chains without sugar lipid moieties. Both of these options are incorrect. First of all, why? Because cytokines are essentially proteins, they are not lipid molecules, and they are released by few different cells in the body if they get an activating stimulus. The incorrect part in this is it's not a small lipid molecule, these are essentially proteins. Similarly, cytokines in their active forms are always pure peptide chains. No, these are not peptides, these are protein molecules. So if you read through this slide, you'll get to know that they are small proteins, but they are not so small that we can call them a peptide chain. They are proteins, they have their proper structure, and these are released by various cells in the body, as I said, usually in response to an activating stimulus. And the we can go through all of these options one by one. So second was cytokines can act in an autocrine manner, so that it affects the same cells that release the cytokines. So the way these cytokines act, it could be autocrine. Autocrine meaning the cell which has released the cytokine then in turn affect the same cell from where it is being released to then change the behavior of the cell. This is very much possible. They also act in a paracrine manner, meaning they'll one cell is going to release the cytokines that will then go and change the behavior of the neighboring cell. It can affect the neighboring cell. This behavior is known as paracrine. And also some cytokines are capable of traveling through the blood vessel. In that case, they will then affect the cells, the behavior of the cells which are very far from the cell that has originated the cytokine. So this is known as endocrine manner. So if you see, it can act as an autocrine, it can act in an autocrine fashion, it can act in a paracrine fashion, and there's some cytokines can have enhanced bioactivity when conjugated with molecules like PEG. This is also possible. And then the incorrect one was the last one. This is also possible. Cytokines are highly potent proteins that bind to their cognate receptors and induce downstream signaling cascades in the respective cells. That is the basically the function of cytokines. So the incorrect part is they're small lipid molecules and they're pure peptide chains. These two options are incorrect. Is this clear? If yes, I'll move on towards the next question. So the next is inflammation facilitates the onset of extravasation, and we have to select two correct options which then describes this process of extravasation. So if you have studied about inflammation in detail, you must know what extravasation is, and then it will be easier to answer this question. If not, then we'll discuss.
Ma'am, option 5 and option 1.
One is it refers to the process of macrophages clearing the dead cells in the vicinity of damaged tissue, and 3. During the process, velocity of blood flow decreases and allows cells to interact with endothelium. Okay, thanks for answering. Anybody else wants to give it a try? No? Okay, let's look at the answer. So the correct option, actually there's some mistake, printing mistake with this particular question. So there are actually three different correct options in this particular question. The first is during the process, the velocity of blood flow decreases and allows for cells to interact with endothelium. So whoever answered part of your answer was correct. You have got the one correct option. Next was it involves the process of migration of neutrophils through the endothelial layer is also correct, and it is the upregulation of adhesion molecules on endothelial cells followed by the release of chemotactic factors. So this is also correct. So let's look at what extravasation is. So as I said, in inflammation, imagine this is your blood vessel and this is the tissue. So in inflammation, let's say there is an injury in this particular tissue. So your immune cells have to flow from this blood, have to transfer from this blood vessel, from the blood flow of the blood to the tissue. So the way it does it, it undergoes a process known as extravasation. So let's say this is your neutrophil here. So what happens is when so first of all, as I said, there is vasodilation. Whenever inflammation happens, this is nothing but increasing the diameter of this particular vessel. So if you increase the diameter of this vessel, the velocity is going to reduce here. So now the blood is going to flow very slowly over this particular endothelial layer. So this is your endothelial layer. This is the layer which lines your blood vessel. So the cells which are now moving in the blood vessel, since the blood flow has been reduced to a very low speed, the cells, the immune cells can then interact with a lot of these factors which are present on your endothelial cells. So these are nothing but they are your selectins, which are transmembrane proteins which then identify this particular neutrophil layer. So what happens is once they identify, they start interacting with each other. So this is nothing but capture of your immune cells. So let's say the blood is flowing here, a lot of cells are flowing here. So the transmembrane proteins, the function of these selectins is to catch the particular immune cell that is flowing in the blood. So once they catch it, once they start interacting, they will then start releasing a lot of factors. Your endothelial cells will start releasing a lot of factors. So these are known as adhesion molecules, the one that interacts with the receptors present on the neutrophils. So your endothelial cell is now going to release a lot of factors. So one of these is MMPs. MMPs are metalloproteins, matrix metalloproteins. So what they do is they start cleaving the ECM that is present in these two endothelial cells, because your neutrophil needs to have space to go from this particular layer into the tissue. So your MMPs will start degrading the ECM a little here, they'll start making space, and then your neutrophils will then be squeezed in from this, and then it can freely enter the tissue layer that is present here, and then it can act against the injury or the pathogen that is present here. So this entire process of capturing of a neutrophil through, and then its entry into the tissue is known as extravasation. So now that you know this, it will be easier to look through the options again. So it refers to the process of macrophages clearing the dead cells in the vicinity of damaged tissue, which is incorrect. That is not what extravasation is. Extravasation is just the process of the entry of your immune cells from the blood vessel to the tissue. It refers to the process of phagocytosis of pathogens that would have entered the body through an open wound. No, this is not extravasation. It is the upregulation of adhesion molecules on endothelial cells followed by the release of chemotactic factors is actually correct. So these, as I said, these adhesion molecules in the process of extravasation, because of a lot of factors getting these endothelial cells getting activated, they will then increase the amount of the adhesion molecules that are there here, so that they can better capture these neutrophils or macrophages. So this is a correct option. Extravasation is the partitioning of cells into different blood vessels based on concentration gradient of chemoattractant. No, this is incorrect. During the process, velocity of blood flow decreases and allows for cells to interact with endothelium. This is correct. It involves the process of migration of neutrophils through the endothelial layer. So if you go over the process of extravasation again from the slides, you'll then be able to read through these options again and you will get the correct answer. Is this clear? Please let me know if this is becoming too fast or too confusing. I can try simplifying it. If there are no doubts in this question, we'll move on towards the next one. The next question is, what is the role of platelets during blood clotting? Again, two options are correct. So read through the options and let me know if anybody knows the answer. Anyone? No? Okay. The correct answer is it stimulates the clotting of blood and wound healing, and it releases variety of chemical mediators that promote clotting and healing of a damaged tissue. So as I said, if there is a blood clot in the body, your platelets are the first cells that are going to repair this particular uh, injury to the capillary. So there are various functions of these platelets. So platelets essentially they stimulate the clotting of the blood and wound healing. They are very important for wound healing, and they release variety of chemical mediators that then promote clotting and healing of the damaged tissue. So rest of the things are not done by platelets. They don't form formation of a clot by they don't help in formation of the clot. They actually help in resolving the clot. And then transport of oxygen, no, that is not the role of platelets. Generation of pro-inflammatory molecules or pro-fibrosis molecules or formation of giant macrophages, these are not the functions of platelets. They are only involved in wound healing. Clear? Okay, I'll move on towards the next question then. Next is, we wish to generate an osteoconductive implant by coating it with fibronectin. For this purpose, an aqueous solution of fibronectin was prepared. Assuming the surface of the implant is positively charged, how should we change the pH of the solution so that a large amount of fibronectin is attached to the surface? And we have the value of the isoelectric point of fibronectin, which is 5.75. And then we have different options here. So we essentially have to choose a particular pH so that most amount of this fibronectin then gets coated over our particular implant. So if you know the concept of pH and isoelectric point, I think this should be easier to answer. Does anybody want to give it a try? Any volunteers?
Option 3, ma'am.
Okay, option 3 is you make your pH more than 5.75 at 25 degrees Celsius. Okay. Anybody else? Thank you for answering. Okay, let's look at the answer. The correct answer is you have to make your pH more than 5.75 but at 37 degrees Celsius. So you were partly right in your answer. But the option here to choose here is you make your pH more than 5.75 at 37 degrees Celsius. So let's look at this. So what we have been given is we have an implant and we want to coat that implant with fibronectin. So and the implant is positively charged. So if you want maximum coating of fibronectin to happen on your implant, since your implant is positively charged, you want your fibronectin to be negatively charged. So and we have been given the pi of fibronectin. So we know this concept that below your pi, if your pH is below your pi, it is going to be the molecule is going to be positively charged. If your pH is above your isoelectric point, the molecule is going to be negatively charged. So to have negatively charged fibronectin, we need the pH more than the isoelectric point of fibronectin. So that makes it clear that we need pH more than 5.75. And since we are going to put this implant in the patient's body, we want the fibronectin still to be coated on the implant even after it is implanted in the patient's body. And the human body is going to be at 37 degrees Celsius. So we have to coat fibronectin to achieve most amount of coating of fibronectin on the implant. We need pH of more than 5.75 and we need the temperature to be at 37 degree. Is this clear? I hope the explanation is making sense. If anybody is not getting this, I can go over this again later on after we finish the entire assignment. So for now, I'll move on towards the next question. So the next is, which of the following is not a physiological event occurring during inflammation? And we have two options to choose from, two correct options to choose from these. So we have been talking about inflammation a lot, so this should be easy. Anybody wants to give it a try? Again, look at the question carefully. We have to select what is not happening during inflammation.
Option 5 and option 8.
1, 2, 3. 5 is reduction in temperature at the site of inflammation, migration of immune cells away from the site of inflammation. Yeah, you're perfectly correct. So why these 2 points are correct, you can go through all the options first. Vasodilation definitely occurs in inflammation. There is increase in capillary permeability, as we saw a few slides back. An influx of protein rich fluid and cells at the site of inflammation, this is known as edema, that happens. Swelling also happens. The reduction in temperature doesn't happen because whenever you have inflammation, the site at which inflammation occurs is going to be a little warmer in temperature, so reduction of temperature doesn't happen. Pain at the site of inflammation will be there. An influx of leukocytes and phagocytic cells into the tissue, this also happens. Migration of immune cells away from the site of inflammation is false, because your immune cells will come towards the site of inflammation because of your cytokines being released there. So the things that don't happen during inflammation are reduction in temperature and migration of immune cells away from the site. Clear? Okay, then let's move on towards the last question of the assignment. It is, which of the following is not an action of the complement system? Again, there are two correct options in this. So for this, you should know what are the functions of complement system, then it becomes easier to answer this question. Anyone?
Option
Yes. Option
2 and 7.
2 is this, repel infiltrating leukocytes from the site of infection or inflammation and cause lysis of gram-negative bacteria in human cells displaying foreign epitopes. Yeah, you're correct. So, okay, no, wait, we are not correct with this. Project foreign objects or organisms for degradation. Yeah, so we have to choose what are not the actions of complement system. So you were partly right in selecting option 2. It does not repel infiltrating leukocytes from the site of infection. It actually it actually signals the leukocytes to come at the site of infection or inflammation. And it does not protect object or organisms for degradation. It actually highlights the organism to be degraded by the immune system. So yeah, so this is actually a function of complement system, where it causes lysis of gram-negative bacteria and any human cells that are displaying foreign epitopes. So if you want, there's another slide that I've included in the presentation, which then highlights the function of the complement systems. Is this clear? So these are all the questions that I had for you for this assignment. So we have reached the end of this assignment. If you want me to go over few of these questions again, or want me to discuss any other concepts, then we can go through this. If not, then we are at the end of the session. Then we can meet next week for week 10 session if there's nothing else to be discussed. I'll be uploading this presentation as well as this video on YouTube. So if you have missed anything from the start, you can go through this. Anybody has anything else to discuss? If not, then I'll stop the meeting here. Thank you all for joining.
Thank you, ma'am.
Thank you. Thank you.